Okay, in this video, we're going to talk about the inherent problem that comes up when trying to replicate the ends of linear chromosomes. So let's take a look at this figure here. If we take a look at the end of a linear chromosome, what we see, of course, one of the strands is going to be the 5' end, and one of the strands is going to be the 3' end. And if we are looking at replicating the strand that is reverse complementary to that of the one that ends at the 5' end here, so this guy here in light blue, that's the leading strand. That's the strand that is being synthesized from the 5' to the 3' at the arrow here. And synthesizing the leading strand to the end of the chromosome does not present a problem, as you can see here. However, it's that lagging strand that is going to be difficult, right? Where we put a primer here and then go away from the end. Lay down a primer here and then replicate away from the end. Put a primer down here, replicate away from the end, and so on and so forth. So by the time we reach the end and we lay down that final RNA primer right there as at the end as we can, so we are quite literally looking right here is right here. There is no more DNA to the left here. We're at the end of the chromosome. We've got ourselves an RNA primer there. Great, beautiful. We've got something that we can make an Okazaki fragment off the end of. Great, no problem. But here's the problem. So remember, the next step is that DNA polymerase 1 is going to remove the RNA, and it is supposed to then replace it with DNA. But remember, what it uses as a primer is the 3' end of the upstream Okazaki fragment. Well, look at this. There is no upstream Okazaki fragment. So the DNA polymerase to replace this primer here has nothing to add to, which means this gap is a permanent gap. Every time DNA replication occurs, it is going to shorten by about the size of a primer from both ends of the chromosome. There's no way around that. Okay, we're going to look at a workaround, but it doesn't actually stop this from happening. So what is that workaround? Well, very early in embryonic development, in all of your cells, at the end of each chromosome is added a series of repeated sequences. TTAGG, 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 and so on and so forth. Now, the enzyme that adds this, right, this isn't encoded by a gene or anything like this. This is added by a special enzyme called telomerase. And telomerase is a ribonucleoprotein, so it's made up of both a protein and an RNA. The protein component is called human telomerase reverse transcriptase, so HTERT. And the RNA component is called human telomerase RNA component, HTERT. Now, the protein component has this enzymatic capability that is called a reverse transcriptase. And what that means is that it uses RNA as a template to write DNA. What RNA template does it use? Its own. The RNA component that is made up of telomerase. So what we have here is the RNA component provides a template. The protein component provides the enzymatic capability, the reverse transcriptase activity. And that what happens here is at the end of each chromosome, there is already a previously established repeat, telomeric repeats. And so they are bound by the telomerase RNA component, like this. That leaves us an overhang. That overhang is read by the protein component and used to make a new DNA repeat, telomeric repeat. And then the whole enzyme picks up, shifts over, and it does it again. So early on in embryonic development, our telomerase is activated, it binds to the ends of all the chromosomes, and it adds these telomeric repeats to the ends of the chromosomes. Now, why is that important? Well, now, when a primer sits, right, on the end of a chromosome, it is still lost, right? Still cannot be replaced. But what is lost is nothing more than some telomeric repeat. It's junk DNA, okay? Which means that there is a certain amount of shortening that all chromosomes can tolerate before they get too short, before you start getting into regions that are not telomeric repeats. Another important feature of these telomeric repeats is that they form this structure called a T-loop, where the free 5' and free 3' ends are bound by proteins and involved in this loop, which means they are no longer substrates for exonucleases. And there's lots of these exonucleases within a nucleus. Their job is to get rid of like introns, for example, that get spliced out. Well, if the 3' and 5' ends of a chromosome were exposed, they would be the target of these exonucleases, or they would be the target of proteins that try to repair double-stranded breaks. Okay, and so these are spared that being accidental substrates by being involved in this T-loop. So telomeres are important for the protection of the ends of chromosomes. They provide sort of a buffer for every time that DNA is replicated. It does still get shorter, but what you're losing is just telomeric repeats and not important sequences. But what that does mean is that it is possible for a chromosome to be replicated so many times that you get to the point where, the cell detects that you cannot replicate anymore. We don't have enough telomeric sequence left. We have to shut things down. And that is called the Hayflick limit. The number of times a cell can divide before it shuts itself down, before the telomeres are so depleted that any more replication would start to hurt the chromosome itself. So let's take a look at that. There's an experiment that demonstrates this, okay? And the idea here is that because somatic cells, so most of your cells, don't express telomerase anymore after that very brief period during your embryonic development. So let's take a look. What we know is that the RNA component is expressed in all cells pretty much all the time, but it's the protein component with the enzymatic capability that is turned off very, very early in development. And so what some researchers had done back in 1998 is they first did a control experiment. They said, okay, let's take a cell, just a fibroblast cell from an individual. Let's transfect it with an empty vector. So this is our negative control. The vector does not have this insert, the insert of the hTERT gene. They transfected that fibroblast cells with that empty vector, and then they added growth factors and forced the cell to divide over and over and over again. So first they showed that nope, doesn't have telomerase activity, right? They did not successfully reactivate telomerase. Good, because it's a negative control. What they saw then with each division, yes, there were telomeres being lost with each cell division, as is what occurs normally. And then what they found is that after about 20 cell divisions, so they were able to force this cell to divide about 20 times, and after that, bang, the cell reaches what's called senescence. No matter how many growth factors you add to it, it simply will not divide any longer, and it reached that. So all of what you see here is what normally occurs to human cells past embryonic development. Now, they took the same type of cells and they transfected them now with an expression vector that contains a gene that, right, the hTERT gene, under a promoter that is going to be active. So they found that yes, there is now telomerase activity within these cells. They found that that there is no telomeric loss. Every time that cell divides, you've got that telomerase that's still active and adding more telomeres. They see that the cell proliferation is what we've referred to as immortal. As long as you keep adding nutrients and adding growth factors to that media that the cell is being incubated in, it's going to divide, and it never reaches a state of senescence. Okay, so that means that it's the lack of telomerase activity that's causes senescence. And what we believe now and what we know now, that senescence is one of the major reasons for the phenotypes of aging. Okay, so as individuals get older and older and older, there is a set of symptoms that happens to everybody as they age. Progressive tissue atrophy, stem cell depletion, organ system failure, impaired tissue injury response. All of this is now believed to be due, at least in part, to this idea of senescence, that most of your cells of your body are going to have a limited number of times that they can divide before they run out of telomeres and therefore stop dividing, thereby causing all of these symptoms. Which means two possible things. Number one, is it possible that the length of telomeres that you're born with will determine just how long you can live? That maybe some families have much longer telomeres and so will on average live longer than families with shorter telomeres? I don't know. That's the hypothesis we're working with here. And the next question is, okay, if that's the case, what happens if we artificially shorten telomeres? What happens if we artificially lengthen telomeres? Another important question is it possible for these age-related phenotypes to be reversed? Well, there was a really cool experiment done in 2010. And what researchers did is they took a mouse line and they created it such that the gene that encodes TERT, okay, that encodes the protein component of telomerase, was under the control of a new promoter, a promoter that is inducible via a drug. Okay, this is important. So now we have a line of mice where we can artificially turn telomerase on and turn telomerase off. The default state is off unless you feed the mouse with this drug. So this is fantastic, right? So they never give the mouse the drug. So they keep telomerase off and they breed the mice and then offspring. Now in those offspring, as embryos, they're supposed to turn their telomerase on to extend the telomeres, but the researchers don't do that. So now the embryos begin with short telomeres and their telomeres get shorter as they live. And then those adult mice with shorter telomeres are bred to one another. The telomerase is still not turned on, which means their offspring start with even shorter telomeres, and so on and so forth. So then by like the third or fourth generation, mice are being born with very short telomeres, such that now they are aging very prematurely, within 30 to 35 weeks. This is much earlier than they would normally show these symptoms. They've got these short, dysfunctional telomeres. They have increased DNA damage signaling, basically the cell saying, hey, don't divide anymore, we have run out of telomeres. And then they have these systemic degenerative pathologies, these pathologies that I've shown you here. Okay, so now they've got this breed of mice that for the past several generations, they never turned telomerase on, such that now this new generation, they aged prematurely. They're showing these aging phenotypes, and bang, now they feed them the drug. They feed them the drug that turns on telomerase in all of their cells. And after about 4 weeks of being fed that drug that turns on the telomerase in all their cells, now they have reduced apoptosis in testes and intestinal cells. Apoptosis is a programmed cellular death. Reduced atrophy and restoration of testes and spleen size. Increased fecundity, so increased ability to reproduce, increased fertility. Increased median age. So what seems to have happened then is not only did they stop the telomerases, the telomeres from shortening, they actually reversed many of these age-related symptoms that were occurring. So this is a really special experiment. And one person in particular was extremely fascinated with this. Her name is Liz Parrish, and she happens to be lucky in that she is the CEO of a biotech company, BioViva Sciences. And she wasn't patient enough for scientists to research this completely and then take it to the FDA or whoever okays these sorts of experiments in humans. And instead, she and her company developed a virus that when you are infected by the virus, it will essentially infect you with an active telomerase gene. So it'll turn the telomerase on in various cells of your body. And she went to another country, and I don't exactly remember which, maybe Colombia, and she became her own patient zero. She infected herself with these viruses, these retroviruses, that then turned on telomerase within her cells. And sure enough, this is before that gene therapy. Her telomeres were about 6.71 kilobases long. And then a year later, they were about 7.33 kilobases long. And then about another year later, 8.12 kilobases long. And the researchers say that if you take all the people's telomeres and you look at, okay, what age are you? What is the size of your telomeres? It turns out that 8.12 kilobases long is about the size of a typical 30-year-old. Okay, and she, Liz, is significantly older than 30. Now, how is that affecting these various things here? How is that going to affect her going on? We don't know. Unfortunately, she is she's the head of a private company, so they are under no obligation to research their results. And of course, they're going to wait until they get some major findings before researching anything, for the fear of being scooped and having their stuff stolen. Now, why is it that people were reluctant to do this? Why is it that you don't want to turn telomerase on in all cells of your body? Well, can we think of maybe some other cells that telomerase is turned on? Well, if you were to say stem cells, cells whose job it is to fuel and give rise to more cells, like adult stem cells necessary to replenish skin or something like that. Those tend to have telomerase in them. But more importantly, one of the necessary activities that must occur to a cancer cell to become a malignant cancer cell is the reactivation of telomerase. Right? If you don't reactivate telomerase in a tumor, then you'll end up getting division, uncontrolled division, but then it'll eventually stop once you ruin your chromosomes and it's done. So you'll get a benign tumor. But if within that cell you reactivate telomerase, now you're in trouble because that cell can keep dividing and dividing and dividing and dividing. And so the question is, will Liz Parrish come down with some type of cancer? That remains to be seen. I highly recommend go on YouTube and Google her name. All right, in the next lecture, we're going to talk about DNA repair.